Hello, my name is Kathy Morrison, and I will be doing a presentation on iron and its uses and applications. This is in fulfillment of the class Chemistry 520, Concepts of Analytical Chemistry. The instructor is Dr. Kenneth Capps. In the beginning, when the first iron was found, it was found on the earth mantle. But this actually came from the heavens above from meteorites. As the meteorites crashed, the people who found them got the iron out of the ore and used very crude weapons. But in actuality, iron is the most abundant element on the earth crust. The properties of iron are as follows. In the periodic table, it is in group 8, which is transition metals. The atomic number is 26. It is strong. It has a high reactivity, high density. It does have a metallic sheen. It does also have magnetic properties. It is moldability, and it forms various alloys, especially with carbon. And these particular alloys are actually different varying degrees of alloys. In this screen, you see the electron configuration of iron at its ground state. And its configuration is 1s2, 2s2, 2p6, 3s2, 3p6, 4s2, 3d6, and in shorthand, it is argon, 4s2, 3d6. For ferrous ion, or Fe2+, the electron configuration is argon, 3d6, and for ferric iron, which is 3 plus iron, is argon, 3d5. This is occurred because it has reduced itself and lost electrons. In the next picture, we see the energy levels of iron in its ground state. As you can see in the 3D shell, 4 of the 5 shells are completely full. 4 of them have a half magnet, and this is what causes it to be magnetic, and it's called paramagnetism. Fe2+ also has 4 unpaired d electrons. However, its energy level is higher since the 4s shell is empty, and it remains paramagnetic. The reason the 4s shell is empty is because it lost its electrons, and it is an empty shell, and it will fill up the 3D before it does a 4s. For the Fe3+, it is paramagnetic as well, but it possesses 5 unpaired electrons because it has donated 3 electrons. So the 4s shell and one of the 3D shells has been donated. But each cell must be filled first before an electron can be given away. So the sequence of paramagnetism follows this order. Iron in its ground state has less magnetic qualities as the ferrous iron, which has less magnetic qualities as the ferric iron. So the ferric iron has the more magnetic qualities of the 3. In this slide, it is just an overview of the general applications of iron. One of the main constituents of iron is in the construction business because it is used as various metals. They use it as a rebar, which is an alloy of carbon and iron, but is very strong and is used for structural foundation. It is also used as roofing material, insulation material on the inside, and electrical components. It is also used in the process of manufacturing, whether the instrumentations that are used to manufacture products are made of iron or the products that are being made are actually iron type products. It's also used in the travel industry. The metal, iron, in different varying alloys are transformed into planes, trains, and automobiles. And then finally, they're also used on medical instruments. The reason this is because one of the alloys is actually stainless steel. Stainless steel is a very good product that can be used for the sterilization and can be cleaned and sterilized for medical procedures. In the next slide, we will show that there are hazards to the iron ore mining. This is the only way to date that iron can be extracted from the ground on the earth. Due to the mining experience, and because it is either an open faced, which is above ground, or a deep iron mine, the dirt has to be removed in one way or another. If it's removed from the top, you are removing topsoil, which is best for growing plants and promoting the environment around it for animals, insects, and plants. And also, the movement of getting the ore to the mining plant causes air pollution because the trucks transporting it are crushing the dirt below them. Then when they go to the plant and they go and dispose of the iron ore into the plant, it needs to be broken down. And when it's broken down, this also produces particulate matter. Then once it goes and is broken down completely and taken into a furnace blast, it releases some toxic gases such as carbon monoxide, carbon dioxide, and particulate matter that's less than 2.5. And then also it produces slag, which is a waste product that may contain cyanide or sulfur from having to break down the iron from the ore. Now, the particulate matter from this procedure is a respiratory hazard. It can cause many issues with the lungs because it is a fine metal and it's tearing the lung tissue. Also, the carbon dioxide is producing and helping with the green air ozone layer issue. Now, this slide, I'm only going to show two ways for testing iron. There are several ways of doing this in many capabilities, such as HPLC, MSGC, GCGCMS, etc. But these two are particulars because they have something in common. And the two things they have in common is they're fairly easy to use and they're fairly inexpensive, which is good because sometimes, depending on the laboratory and the research that is being conducted, cost and ease of use is something that they cannot afford to get. So in the UV vis spectrophotometry, we are isolating the wavelength for iron. We have an iron source, a light source, which is usually just a deuterium lamp or halide. Then we have an entrance slit, which allows for the light to go through, but it is causing it to concentrate in one area. It will go into a dispersive element, and this dispersive element is usually a prism. And prisms can come in varying shapes and different sizes as well. And as you can tell, it breaks down the color of the light. The next is we have an exit slit. In this exit slit, it is set up where it only allows the wavelength for the iron to come through. And all of this is a monochromator. Next, we'll go through a sample. And in this cuvette, you will have your product, your sample that you are trying to measure. This will be using Beer's law, where you have A for absorbance, B, the length of the cube, and the epsilon, which is the molar absorptivity coefficient of the sample. This then goes and proceeds to the detector. The detector will print out a graph that shows the wavelength and where the iron actually showed up at. The flame photometer has the sample in solution. So anything that needs to be done by flame photometry has to be emulsified in a solution form. It cannot be solid. It then goes through to an air compressor. The air that is being used is an inert gas, so it does not affect the amount of iron that is going to be used, and it is called a carrier gas. It then proceeds into a nebulizer. The nebulizer has this fuel that goes through and causes it to break down and blow up the particles of the iron and any kind of compounds in there. After that, it goes through the flame, and in the flame, it will emit the iron. The iron light source then goes through a monochromator filter, which then is magnified by a lens into a detector and recorder. This also prints out a graph that shows the amount of iron that was in the initial sample. The biggest difference of these two methods is in the UV vis, you can salvage your sample, and it is small sample size. For flame photometry, you need a larger sample size, and it's a one-time use sample. So you have to be very careful in what you will be using it for. Next, iron, even though it's a trace metal in the biological system of human beings, it does a lot of jobs. The iron-dependent activities, for example, the iron sulfur proteins, and the type of function common is iron sulfur proteins and iron sulfur clusters are involved with the mitochondrial substrate metabolism in the TTCA cycle. And it also is DNA in the cells and mitochondria, and it is an initial response to iron depletion and hypoxia or cell death. It also uses dehydrogenases such as isocitrate, NADPH, and succinate. These aid in the citric acid cycle in mitochondria, the serial oxidative processing in ADPH, ATP and GTP, which are sources for energy, and the substrates for synthesis of amino acids, lipogenesis and glycogenesis. So the synthesis of amino acids are the foundations and building blocks of every protein in our body. So that is a big use for iron. And we also have such as cytochromes A, B, and C, and cytochrome C oxidase. These are used in specific for electron transfer chain in mitochondria and its inner membranes, and it oxidizes the NADPH from citric acid cycle to drive the synthesis of ATP. The next two slides, we will be dealing with the biochemistry of iron involved, in particular to neurodegenerative diseases. First, how does the body get iron? The body does not produce its own iron per se, so we must ingest some. And you can get that from animal products or non-animal products such as spinach, which has a high iron content. These two are then ingested into the system. Also, any iron that is in the body is recycled. So that is a good way of reutilizing any iron that you have. You ask what happens when the iron is ingested? The iron-bearing food is taken down into the GI route. And in the lining, the ferric iron is reduced to ferrous iron. The ferrous iron is easily absorbed by the duodenum. When it is absorbed, it is then transferred into the system in various places, and it will be transported by ferroprotein to various organs such as the liver, the brain, or even the skin. And then you ask, why is this important to the neurological system? Well, as we saw from the previous slide of all the iron-dependent abilities, it is also used to build up myelin sheath, transport of oxygen to the brain, DNA synthesis, and energy use. Now, when I say transfer to the brain, that is very important. There's only a few items that can cross that blood-brain barrier, and iron is one of them. In the next slide, we will continue, but on this particular one, it's like, what kind of neurodegenerative diseases are there? Well, we have Alzheimer's disease, dementia, Parkinson's disease, Huntington's disease, amyotrophic lateral disease, ALS, also known as Lou Gehrig's. Now, Huntington's disease and ALS are primarily a genetic disease, but the iron use is helpful in the prognosis of the patient's health. What are the main causes indicating iron issues? Iron overload leads to an oxidative stress, which is a type of cell death, and low iron production leads to nerve sheaths not replicating as well and oxygen not getting to the brain cells, causing hypoxia or cell death. When do these items occur in the life of cycles? Well, in one cycle, it is premature children because they do not have enough circulating red blood cells, and that could have happened during the delivery of the child because there might have been a hemorrhage occurring and the loss of blood before they were able to be on their own. And also because they're premature, there is a possibility that their liver, their spleen, their gastrointestinal route has not fully developed, so they will not be able to participate in the hemoglobin process. The second one is older people. And the reason it does that is because some of the processes slow down. And also because some of the areas where the iron comes in start breaking down. For example, in the GI tract, as we get older, we develop ulcers, we develop gastroenteric reflux disease, heartburn, and because of this, the lining of the GI tract can slowly be sloughed off, inhibiting the absorption process and the breakdown so that the iron cannot go through the system. Also, there is a possibility that it cannot convert the iron and does not allow for that used up iron to be moved out of the system to be recycled. So it will cause either an iron deficient or an iron overload. When you're testing for neurodegenerative disease, you can do blood tests, and some of these blood tests include total iron bonding capacity, ferritin, ceruloplasmin, transferrin, and unbound iron binding capacity. These are tested with a spectrophotometry, but on automated platforms that there are several different types of manufacturers that they make, but it's basically a spectrophotometry type of methodology, whether they use color or a UV-Vis type of method. The second one is the MRI scans. When a person is initially diagnosed or is being worked up, they will take a baseline MRI scan to see what your brain looks like initially. And as your treatment occurs, they will take other MRI scans to see whether there is a breakdown in the brain in accordance to the MRI scans. Next, there might be some brain biopsies, and these will be stained for specific immunochemistry markers. For example, there might be a stain that will be looking for hemosiderin, which is a breakdown of iron, and show that there is an abundance of iron, which is an iron overload issue. Also, the first line of defense should be your general checkups, and it will do the prevalent symptomology such as extreme shakes, memory loss or fog, walking, talking, or focusing, and other general symptoms such as that. And then, of course, finally, the age of the patient. So, in conclusion, although neurodegenerative disease is a scary condition to go through, the best outcome is knowledge. You know your disease and where to get help. Be your own advocate. If you feel that something is occurring, push your doctor to investigate and get answers before they qualify it as you are getting older. And also, do not allow the lack of funds to keep you getting the health care that you deserve. And even though the health care is not at its prime work, it still is there for you to help, and the doctors can continue to help you in that instance. And the final slide is a list of references. So I thank you for your time.